2,600 different projects being done for iGEM, right? And although the projects are basically based on synthetic biology, they tend to have a very interdisciplinary sort of an approach because biology is essentially applied physics, maths, and chemistry, current biology. And apart from the competition, iGEM also has other events. For example, they have different podcasts to where they get some Nobel laureates and other scientists, especially from synthetic biology and various walks of biology, where who explain different cutting edge research that is being done. Every country and even continents have their own iGEM leagues. For example, the Indian League, which is organized by the Indian teams. Here we basically teach students from schools about various molecular biology techniques. We teach them about bioinformatics. We teach them about synthetic biology and its applications and so on. And iGEM also has a very interesting program called EPIC, which is basically they support the entrepreneurship. So if you want to develop your project further beyond just the competition, if you want to make it your career, if you want to form a company, then iGEM can help you with fundraising and such, which through their EPIC program. Okay. So moving on to focusing on the competition here. So the competition has various tracks. So once you're making a particular project, once you have a particular project idea for iGEM, it has to belong to one of the 12 or 13 different tracks. They could be diagnostics. For example, you can make a diagnostic kit. They could be therapeutics. You can make a particular drug maybe for curing a particular illness. It can be related to software. You can just have, say, some sort of a machine learning software which identifies a tumor from a microscopic image. That kind of project has been done in iGEM. It can be a foundational advanced project, which is what we are doing this year. Our project is foundational advanced, where you have to create a sort of a general method or a general platform which has multifaceted applications. What I mean by that is, for example, PCR, right? Polymerase chain reaction. I believe even people, not from the biology background know this because of COVID and all. So PCR is a very general molecular biology technique. It can be applied to detect COVID and a very lot of diseases. It can even be applied in research in various aspects, right? So that would be something which would be part of foundational advanced track. Food and nutrition, you can develop a particular bacteria or fungi which has very high nutritional value, and it can be, I don't know, food for astronauts or something like that. So these are different tracks you can have in iGEM, and your project has to belong to one of these tracks. So what exactly constitutes, what all are the aspects of an iGEM project? First of all, you need to have the project idea. And once you have the project idea, you will have to design experiments, right? Because this is synthetic biology. So there are, biochemistry, chemical biology, pure biology experiments in the wet lab that you need to carry out, that you need to design and carry out. And apart from this wet lab, you also need to have a very extensive dry lab, which basically means computations, simulations, and modeling. It could be mathematical model of the wet lab experiments that you're doing. It could be some sort of a molecular dynamics simulation. It could be some sort of a molecular docking simulation of whatever is, whatever you're working with. So a very extensive dry lab has to be carried out as well, which is where you would need not only people from biology background, but also people who know a bit about computers, some physics, some chemistry, and so on. Then there is a team website. So this website is called the wiki. You need to design a wiki where you need to put everything, all the experiments that you have done, all the dry lab simulations that you have done, and you can look at various wikis. If you search up iGEM team wikis, you'll get a lot of examples. So the wiki design is a very important aspect of iGEM. You need your website to be informative as well as well designed. Then comes human practices. One of the very important central aspects of iGEM happens to be human practices. What it essentially entails is communication with humans, right? With people around you. So it could be, for example, we organized a synthetic biology workshop for school students, right? That comes under education and communication, which in turn comes under human practices. Then there is a integrated human practices, which is also a part of human practices. It basically entails that you need to talk to the stakeholders of your project. So what that means is, for example, you're making a drug, right? So you need to talk with the pharmaceutical companies, you need to talk with the doctors who give you a better idea of how you can improve your project or how you can implement your project in a better way. So these are the aspects of human practice. And then comes fundraising. I believe, I think there are some people from IISER Kolkata who may be, very aware of these things. So yeah, so fundraising, as you know, that part of it is provided by the institute. However, a part of it has to be, you have to get it from private industries. It can also be from the government, like the DBT, DST, and so on. And then there is a presentation you need to make, videos and stuff. So this particular presentation that we are having can be considered under human practices. Okay, we are trying to explain our project and what iGEM is to everybody around here. And then comes the highlight of the competition, which is the Grand Jamboree. It is like a conference. It's like a congregation of all the teams who present their project. This year, it will be held in the Porte de Versailles, which is located in Paris. It's the Paris Expo. So we'll be going there and presenting our project. So that is the Grand Jamboree. It's something really interesting. Get to meet people from all over the world and so on. Yeah, so after iGEM, as I mentioned, that you can make iGEM your career. So there are a lot of different companies, as you can see, companies from so many different aspects, so many different walks that come out of iGEM. You have companies which help you overcome disease. For example, they make drugs, they make diagnostics kits. There are companies which improve water quality, eliminate pollution, improve enhance food production. They even work, or there are companies which work on making science accessible, which is basically human practices. And there are companies and establishments which basically have come out of iGEM projects doing all these things. So I hope I could give you a basic idea of what iGEM is all about. I don't want to waste too much time on this. If everything's fine, if there are no real questions about iGEM as such, then I can move on to our project. But before I go, I just very briefly, something I didn't want to focus on is the awards. Okay, you get a lot of awards for your projects in iGEM. So you will be judged, right? You will be judged in the Grand Jamboree. You will be judged all throughout your project. You'll be told to upload various things and you'll be judged. So there are medals. There are gold, silver, and bronze medals. There are awards for the best project in each track. For example, you could have the best diagnostics project. You could have the best food and nutrition project. You could have the best hardware project, and so on. So there are awards for each tracks. There are also awards for human practices, the best human practices. There are awards for the best wiki, the best website, and so on. So there are a lot of awards, but more than the awards, it's about the experience, I guess. So yeah, it's a cliche, but sort of what we are going at. So if there are no questions, I'll move on to our project this year. All right, then, okay. So since there are no questions, I'll just move on to the specific project that we are sort of doing for iGEM this year. Right. I hope the screen is visible. Moithra, can you confirm? Yeah, great, fantastic. Okay, so we are team IISER Mohali, and this year we are doing a project called NeuraSyn. So a more elaborate title would be Neural Chip-Based Simultaneous Microbe Detection using Aptamers. And Kalyandeep and myself will be presenting this particular project. So before we get into it, I'll just give you a brief idea of how we are going to go. First of all, we are going to explain the different components, the neural chip and the aptamers, and then we'll give you guys a sort of idea of the platform that NeuraSyn is, and how these neural chip and aptamer come together to sort of detect microbes. And then we are going to also brief you a little bit on the sort of work we have done, the dry lab and the wet lab work we have done on this particular project so far. Right. And before going further, I would like to introduce you to our mascot this year, who happens to be Monsieur Neurapar. And then I'll hand over the mic to Kalyandeep to sort of explain what a neural chip is.
Hello, am I audible? If anyone could confirm. So we'll start with what a basic neural chip is, and then we'll look into some properties of neurons that make a neural chip possible. So a neural chip is basically like a computational device, and it's not a potato chip. So if anyone's hungry,
it's his attempt at a pour as well.
yeah. So if anyone's hungry, a neural chip will not satiate you. It's made out of neurons, as the name suggests, and it's basically, it arises from a property that neurons can be excitatory or inhibitory, and they can communicate in a grid using these electrochemical signals. So these are the two properties that give an emergent property of computation that the neural chip can do. Right. And what exactly are these connectivities, or what exactly is thinking according to chip? So what I mean when I say thinking, it means that the neural network can form micro network motifs, and these motifs can evolve over time, right? So that is what thinking means. So thinking is intricately connected to learning. So when you learn activity, it is corresponding to a certain micronetwork motif on the neural chip. And what is a micronetwork motif? It is basically a connection of neurons. So as you can see, there are some very common micronetwork motif, that is feed forward excitation, lateral inhibition, or recurrent inhibition. So these are very common micronetwork motifs, but there can be much more complicated micronetwork motifs. And basically every state, every input and output goes through a series of micronetwork motifs. And because these motifs can change with given conditions and changing in the parameters, that is what thinking means according to this chip. Like, that's how we define thinking in terms of the chip. So how is a neural chip? So basically, the way a neural chip thinks is that their connection patterns can change, which is the definition of thinking. And why do their connection patterns change? Their connection patterns changes because of something called as Hebbian plasticity. So that will be explained later. But what it basically implies for our project is that if the chip is presented with data and big air quotes data, it can decide whether the data is significant enough to make a certain judgment. So it can make a judgment, like a artificial neural network. Now, exactly what do I mean by data? So nerves take in electrical and chemical signals, but electrical signals are much faster, like, and they're easier to modulate than chemical signals as an input to neurons. So that is our data. We'll be taking data from a real life assembly of aptamers, and the method will be explained shortly. But in brief, we'll be taking electrical data from aptamer and just sending that to our neural chip, and the neural chip will be pre-trained so that it can look at patterns in the data. And now, how do you train the chip? It is trained by a data set which is generated by experiments on the aptamers. And there's a basic threshold, it identifies a soft threshold. So now, has it been done before? Yes. So this is a picture of MEA60, which is a commercially available chip which is used to record neural data. uh, MEA basically stands for multiple electrode array, and 60 I think stands for the number of electrodes present on the array. But what we are trying to do is, because our data is not exactly multiple location, like the spatial location of the neurons don't really matter for the data that we are trying to classify, we can make a much more simplified version of MEA60 that still generates a much more so this generates a lot of noise. We considered this pattern, but this generates a lot of noise. And if we can use less number of electrodes for the measurement, the noise factor sort of cancels out. So our design is based off of this, but not exactly. Now, how do you train the neural chip? So when you have a correct output, so I'll just go into a bit of Hebbian plasticity. Hebbian plasticity, in the simplest of its form, you can think about two neurons. And if the two neurons fire together, their connection weightage increases. And if they do not fire together beyond a certain threshold of time, then their synaptic connections are weakened. So you can monthify this and call it as neurons that fire together, wire together, neurons that fire apart, wire apart, right? So when you have a correct input and your in signal, so your signal in is a constant voltage of AC, and then there's no change because neurons usually react to changes in the electrical current. Now, if your current isn't changing, if it's a constant AC voltage, it's just a constant background. So your neurons won't really get trained on that. The output, however, if it's a correct output, then we decrease the voltage, but we increase the frequency. and the reason we increase the frequency is that once you increase the frequency between two neurons, they wire together because they are firing together at a certain region of frequency, at a certain high region of frequency, and the result is that the connections are strengthened. However, if the output is wrong, then again you just do the opposite. You give a higher voltage and at a lower frequency, and that just increases the probability that the neurons will fire apart, and thus it will weaken the connections. If there are any questions regarding this part, you can stop me at any point. Now, why do we use a neural chip? Because neurons are faster to train. So, because neurons are something called as loosely connected networks, so it's basically like an echo state network, and because it's loosely connected, there are a lot of connections that you don't need to consider of the get go. Secondly, faster in the sense neurons can be more plastic than some other software generated neural chips. And the second and the third most important reason is that uh, the loss of information, that is the cell to pixel problem, is solved. So, what do I mean by that? Every time.
Yeah, just a second. There's a question. There was something about why do we use a high voltage and a low frequency one. Can you just explain this part?
Yes, so actually we use a high voltage and a lower frequency because if you go through the equations of two neurons in a single line, then and I mean this all comes from the realm of probability. But at conditions of high voltage and low frequency, the probability that the neurons will fire apart is higher, and at conditions of low voltage and high frequency, the probability that the neuron will fire together is higher. So then, going back to our Hebbian plasticity paradigm, we derive that that should be the general case. But this is important to remember. We are not changing the connections at an individual neuron level. We are doing it more at a global level because your inputs and outputs are sort of processed at a global level. So there's a global decrease in the synaptic weights and there's a global increase. So that's the sort of way we'll be working with these sort of the training of these chips. Right. Did that answer your question?
Yeah, I think Orkojyoti, you asked this question.
Yes, 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 it's okay.
This answers your question, or I
Yes,
mean,
yes, it's okay.
okay, great,
Yeah.
fantastic. I think we can continue.
Yeah. So as I was talking about the cell to pixel problem, the cell to pixel problem is basically what happens is that when you're trying to analyze a certain image, you lose a lot of the data because the data that you're taking, maybe you're taking a picture. A pixel is a average value of a certain amount of area. So everything in that area gets reduced to a single number. So therefore, obviously, you are losing information. Also, the thing that happens with pixel is that maybe the bacteria is present in half of the pixel in the picture, and the other half of it is just empty space. So what will happen is that uh, the pixel will average out the bacteria and the empty space into a single pixel, right? So you are going to make changes to your original data set. But with neurons, that is not the case. Neurons will not detect a signal when there is no signal, and there's no averaging it out as such. I mean, yes, it will take a certain number to fire, make the neuron fire, but there is no averaging out of the get go. And also the sustainability problem because they don't have any like real carbon emission to speak of. Neurons are reusable and biodegradable, and also the fact that you can really integrate biocomputing with daily life. You can integrate them with real biological matter. That sort of helps us to have a technological leap into the future.
Right. Basically, sustainability aspect, I'll come back to it once again when talking about the motivation for a project. So I believe the neural chip was, got at least a basic idea of what we are trying to do with the neural chip. If there are questions, you can ask, because then we'll be moving to Aptamers, a new topic. Are there any questions? I mean, this is an informal discussion, guys. There is, I mean, you can definitely ask us. Okay, if there are no questions, then we'll move on to aptamers. Okay, so what are aptamers? So aptamers, as I had mentioned in the abstract of the talk, that they are oligonucleotides, and that still stands, obviously. So they are single-stranded oligonucleotides, and they are not always single-stranded, mostly single-stranded. They can be double-stranded also. So what oligonucleotides basically means is that I believe that most of you know what a nucleotide is. Obviously, it's just high school knowledge. But so when you string together quite a few nucleotides, not so much that you can make a nucleic acid, not so few that it doesn't really make sense or make a difference, is what you get right in the middle is called an oligonucleotide. Oligo basically means few, right? And a nucleotide, just to summarize, is basically a nitrogenous base, a purine or a pyrimidine base attached to a ribose or a deoxyribose sugar, which is in turn attached to a phosphate group, right? They are DNA or RNA, and as I mentioned, they're oligo, which means that they are around 15 to 60 bases, right? 15 to 60 bases. However, the aptamers we are using are slightly longer. They're around 80 to 90 bases, but they still this come under the realm of oligonucleotides. So the feature of aptamers that we plan to use in our project is that their ability to bind to various targets. And those targets could be small molecules such as vitamins, they could be drugs, they could be macromolecules such as proteins, carbohydrates, glycoproteins, and even lipids sometimes. They can be viruses, or they can even be cells such as bacteria and even mammalian cells. Okay, so the aptamers go and bind to these targets, and this is the particular feature of aptamers that we'll be using in our project. And the binding is through hydrogen bonding, electrostatic interactions, hydrophobic interactions such as van der Waals interactions, and stuff like pi stacking interactions. Right. So this particular schematic sort of gives you an idea of how an aptamer binds. So we have an aptamer sequence, and as you can guess, it's a DNA sequence because there is thymine. So this is the single-stranded aptamer sequence, and then it undergoes a 3D structure formation, right? Similar to a protein folding, right? You can imagine it like a protein folding. It forms a functional aptamer. It folds and it forms a functional aptamer, and this functional aptamer goes and binds to the target. In this particular case, the target is the biomarker, which is usually a protein or a glycoprotein, which is present on a cell. So once it binds to the target, there is some physical or physicochemical or chemical change which sort of indicates that the aptamer has bound to the target, which in turn tells you that this target cell or this target biomarker is present in your sample. Okay, and I'll be explaining how exactly that works. But let's look at how we can use this fact that aptamers can bind to various biomolecules or cells. Well, aptamers can be used for detection of various substances. They can be bacteria, they can be viruses, they can be even toxic substances, they can be drugs in your body, they can be vitamins and whatnot. And they can also be used as therapeutics. How exactly? Well, if an aptamer is binding to a particular protein, if that protein is important, say the spike glycoprotein of the SARS-CoV-2 virus, right? I believe you guys know about the spike glycoprotein. So if the aptamer goes and blocks or binds to the spike glycoprotein, then the virus does not get entry to the cell. So in this particular case, the aptamer prevents infection. So the aptamer is acting as a therapeutic. However, in our particular scenario, we'll be using it for detection of substances. Another feature is that the aptamer is highly sensitive. It can detect very few molecules in the sample. It is also highly specific. What that means is that if you develop an aptamer for, say, substance A, the aptamer which has target A, it has very little cross reactivity. It won't very easily go and bind to substance B. Okay, it's specific to substance A. And then a very interesting feature, I personally found it pretty interesting, is that it is independent of immunogenicity. What immunogenicity means is that the ability of a particular pathogen or substance to induce an immune response. Right, so different bacteria or different viruses have different immunogenicities. They induce different levels of immune response. Some induce a high immune response, some induce lower immune responses. So what happens in case of antibody detection? For example, ELISA. You must have heard of ELISA, for testing dengue and various other diseases. So what happens in ELISA? They depend on antibody detection. So these antibodies are produced in response to a particular pathogen, right? So there could be a situation where too much antibodies are produced, whereas the amount of pathogen or the number of pathogen is very little. Okay, it does not even count as an infection. However, you have a huge antibody production. An opposite scenario can also happen where you have a lot of pathogens in your blood or your body fluids, but very little antibodies are being produced. And also another factor is that the number of antibodies are limited, right? Number of antibodies are five, but the number of pathogens are huge. So when you're relying on antibody for detection, the specificity kind of reduces, right? You get a lot of, even the sensitivity reduces because you get a lot of false positives and false negatives. However, since aptamers are directly binding to the pathogen, you don't need to worry about how much of an immune response that pathogen is producing. So aptamers have this particular advantage over antibodies, and also they are comparatively less expensive when compared to antibodies. So moving on to how exactly the detection happens using aptamers. So aptamers are placed on a reduced graphene oxide sheet, right? So I believe everybody here has an idea what graphene oxide is. So graphene is just a layer of a graphite which has like 6 carbons in hexagonal shapes, and then you oxidize that and then you reduce it, right? So you have a reduced graphene oxide sheet which acts as a very good electrode. Okay, and we sort of place our aptamers or fix our aptamers onto this reduced graphene oxide sheet. Now the fixing can simply be dropping off the aptamer. Even then you will have some sort of binding. The aptamer will stay on the surface unless you like wash it off rigorously. But normally chemical fixation is done. So what that means is that you modify this reduced graphene oxide sheet, you do various levels of modification so that your aptamer can bind better, right? Okay, so once your aptamer has bound to the graphene oxide sheet, then you put your sample, and your sample contains your biomarker or your cell or whatever target molecule you want the aptamer to detect. And you can see in this particular image that the biomarker comes and sits on the aptamer, or rather the aptamer binds to the biomarker, and this particular binding causes a change in impedance or resistance. More specifically, it causes an increase in the impedance or resistance of the system. As I mentioned, that you need to have a physical parameter to understand that the aptamer is bound. In this case, it is the increase in electrical impedance or resistance. And how do we detect this increase in impedance? We do something called electrochemical impedance spectroscopy, or EIS, right? And after doing the EIS, you get this kind of a plot, this kind of a graph, which is called a Nyquist plot. And if you look closely here, the black squares, the tiny black squares, which is basically the baseline. Here, this indicates the impedance when it is just the aptamer on the graphene sheet. There is no target, no sample involved, right? And as you gradually introduce more and more concentrated solutions of sample, there is an increase in the impedance, which sort of tells you that the aptamer, more and more aptamers are binding to more and more target molecules or target cells. And as the concentration of the cell of the cell culture or the solution increases, for the increases the impedance increases further, right? So it gives you an idea that more the increase in impedance, more the number of target molecules are present in your solution. Okay, this is I believe very quite clear from this particular graph. So if you have understood the basics of the neural chip and the aptamers and how we are using the aptamers, it's just before I forget. So Kalyandeep mentioned generating a database, right? Kalyandeep mentioned generating a database to train the neural chip. So the database is generated like this. The database has to be generated using electrochemical impedance spectroscopy. So for example, we have, say, a bacterial culture of a particular concentration. We put that onto our aptamers and we check the impedance change, and then we change the concentration of the bacterial culture, and then we put that onto the aptamer. So we basically get a data set of the impedance change corresponding to different bacterial concentrations or different microbial concentrations, right? And that is our data set which we feed to the neural chip, which we train the neural chip using. So the neural chip gets an idea that, okay, if this is the impedance change, such is the concentration of microbes in the sample. Right? This is how the database generation is done. Okay. So once the neural chip and aptamer are clear, the concept of the neural chip and aptamer are clear, and please go ahead and ask questions if it is not clear. If any part is, if you have any doubts, okay, then we can sort of bring these two together and move on to what our platform NeuraSyn is all about. Absolutely, absolutely. Please go ahead. Ye- yeah. So actually, this particular and this actually had intrigued me as well. This particular, sort of bell part or this dome is not exactly something that is taken into account. Okay, although you get such a behavior, uh, when you see the impedance plots, it's a linear increase. So if you ignore this dome, right? If you ignore the dome, you get linear increase, right? Right. So they have plotted this dome, but in various papers, you will actually get to see a linear increase of impedance corresponding to the increase in concentration. And I'll just come back to that, because this feature of linear, linear relation or linear dependence of impedance increase is what we use in our project as well, right? Okay, but your question, yeah, this particular thing, I am not very sure that there is a very clear explanation as to why there's an initial increase and then there is a decrease. And also, it's also about how a Nyquist plot is, being done. Because if you have the plot parameters are slightly different, you might get a slightly different curve. Okay. That's one more aspect, right? Okay, so moving on to the motivation of a project, what sort of motivated, and by this time you have a bit of an idea. So we want to make a detection kit, right? The neural chip is part of a detection kit. Why do we need a detection kit? Well, because current methods of detection, such as cell culture, are very slow. They take approximately 2 days. And if you have something serious, for example, if you're affected with some serious bacterial infection or any infection for that matter, by 2 days, you could go into some shock, septic shock. You can even die. Okay, that's unfortunate that a lot of deaths happen due to delay in detection, and cell culture is currently like the gold standard of detection of bacterial infections. So cell culture method is slow. That's point number one. Next point is that fluorescence based detection, you guys must have heard about, using antibody fluorescence methods. Even aptamers can be coupled with fluorescence to have fluorescence detection systems, but it results in a high false positive because of spectrometer problems, because of system instability. So what happens is that if spectrometers are normally too sensitive, so they will give you a lot of false positive values if you're doing fluorescence spectroscopy. And with microscopy, it's difficult because you will get certain amount of fluorescence no matter what. Okay, and it's difficult to quantify, what you will call positive and what you will not call positive, right? So that's a problem with the fluorescence based detection. Anything to add?
I would just add one more point regarding the system instability part. So, what do I mean by system instability, especially when fluorescence is such a research and such a well established field, especially in detection? So, system instability refers to the amount of variation that a molecule has, basically the RMSD values, when a fluorescence and when a fluorophore quencher system is attached to it. So now, when we speak about fluorescence, we typically think of ELISA, things like ELISA. But what we must remember is that ELISA is a huge protein. Compared to that, aptamers are tiny, and aptamers are very flimsy as molecules because you can think of it just as a single strand of DNA. So it has a lot of motion, and then when you attach heavy weights to the ends, there is a high chance that the fluorophore and the quencher will, even after you cut the aptamer, maybe the fluorophore and the quencher will stay connected. That is why the system instability in aptamer based fluorescence detection is high.
Right. Yeah. So that covers it. And next, we'll take sort of a jump onto a different. Yeah. Okay. So as Mithra, I hope this answers your question, kind of. It's related to your question that we use the linear increase of impedance with increase in cell concentration as because it's linear. That really helps us to compute, because linearity is easy to simulate. Linearity is easy to predict. Right. Okay. Now we sort of take a jump onto a different aspect, sustainable computing, that Kalyandeep mentioned. And here I want to, sort of talk a bit in detail. So we are all using laptops or maybe mobile phones or maybe, sort of desktops to listen to this talk and in our everyday lives. And as we might have read a lot of factoids in newspapers and, general knowledge facts, that it causes a huge amount of carbon emission, right? So for example, I read somewhere that if you send, for every thank you email you send, there is a certain amount of carbon emission, and every person, if a person who is, actively involved in emailing, can result in kgs of carbon emissions just by thank you emails. So they were like, just stop sending thank you emails. There is no point doing that. So such carbon emissions is a real problem even in home PCs, right? However, it is an even bigger problem when you go to high performance clusters, which are GPU clusters, right? Doing high level of computations, for example, mining Bitcoins or predicting the weather, doing space research. They have very, very high amount of carbon emissions. And even in supercomputers, you have a huge amount of carbon emissions. Another aspect another problem to modern day computing is the heat generated, right? When you're running a very sort of extensive program, or I would say intensive computing intensive program on your laptop, it heats up, right? And you also must have noticed that the server rooms in your institutes, maybe, or if you've been to one, are placed in a really cold chamber, right? The AC is running in like 18 or even lower, right? So the problem is there's a huge amount of heat generated. So that also contributes to global warming, right? So there's carbon emission, there's heat generated. And last but not the least, the silicon chips that we are using in our computers, the mining of silicon itself is a very environmentally polluting process. So it consumes a lot of energy, and it also pollutes the environment when you're mining silicon. So sustainable computing is a need of the hour. And the neural chip, as you have probably guessed by now, since we are using biodegradable, eco-friendly cells, neurons at the end of the cells, for computation as computational entities, it really reduces your carbon emission. It really reduces your heat generation to a great extent. And another aspect is the disposal of electronic waste. So some of you might be thinking that, okay, with the Aptamers, why don't you use, instead of neural chip, why don't you use a similar electronic device or some sort of silicon chip? So one problem is all these things, but in our particular aspect, the heat generation and carbon emission will not be so high. But what will be a problem is the waste generation due to the silicon chip or the electronics that we will be using. So you know that there are batteries and there are heavy metals in these batteries. They can leach into the soil, causing environmental pollution, leach into the water, that makes water unfit for drinking, and so on and so forth. So electronic waste generation is another problem we aim to address, right? So with the neural chip, you have neurons. Even if you discard these neurons, these are non-toxic things, these are biodegradable, and the chip is also reusable. It can be reused again and again. And a fascinating aspect of the neural chip, which Kalyan was really excited about, is that if you break a neural chip and if you glue it back together, you can reuse it, which cannot happen with an electron. You can't break your phone into half and glue it back together, expect it to work. But the neurons will rewire, because these are cells, these are dynamic entities. They can rewire, and you just need to train them again, and they can continue working. Okay, maybe not as efficient as earlier, but they can still keep on working pretty well. So that's another aspect. So we are reducing the electronic waste generation. Okay, so moving on to the actual project, what it is all about. So in the project, we aim to detect two different bacteria, Escherichia coli and Salmonella typhimurium, and we also aim to detect a fungus, Penicillium, not necessarily Penicillium chrysogena. We aim to detect the genus Penicillium, which produces penicillin G, or benzyl penicillin. So we cannot directly detect a fungus. I mean, we could not find aptamers which directly bind to the fungi, but we can indirectly detect it by detecting benzyl penicillin, which is produced by Penicillium in a particular sample, right? So that's sort of the one-line goal of our detection platform. And of course, it's faster than existing methods such as culturing. It is basically a zero-time kind of a platform, so it should be able to give you results in a matter of minutes and seconds. Okay, leave alone days. It's much simpler than PCR. This is a very important thing, because in our country, in third world countries like India, there is a huge problem of skilled expertise. Expertise when it comes to doing complex reactions or complex molecular biology stuff like PCR, which was felt during the pandemic, because the number of samples tested in India were very low because of the lack in expertise. So our system happens to be much simpler. It can be used by even meagerly trained personnel. So it does not have, you do not have to know a lot of molecular biology to use our system. So it's much simpler, and it can reach even the remote areas. And applications, as you can probably guess, we want to detect in blood infections, such as septicemia or sepsis. I must remind you that this particular project is a prototype or a proof of concept. Septicemia and sepsis detection might be slightly more complicated because there are a lot more bacteria involved. But if you can demonstrate it for 2 bacteria, it can be expanded to maybe 10 bacteria. It doesn't really matter. So we want to detect septicemia, and what septicemia means is that due to various bacteria infecting your body, you have a systemic infection. You have a systemic infection which overwhelms your immune system, and even in the blood you have presence of a lot of bacteria, and it eventually leads to death. Okay, septicemia is one of the leading causes of death in not only in India, all over the world. Okay, we can also detect food and water contamination. As you might be knowing, that Salmonella typhimurium, causing salmonella poisoning, is causes gastroenterological infections. These basically enter the body through contaminated food and water. Even E. coli can contaminate food and water. And the novelty, as you can probably guess, is biocomputing, is the neural chip. Okay, moving on to this is sort of like a schematic of what our platform is going to look like. You have aptamers on a test strip. So these aptamers, as I mentioned, so there will be three different aptamers for three different targets, which is E. coli, Salmonella typhimurium, and penicillin. These will be fixed onto the reduced graphene oxide sheets. You will be putting the sample into this, and then you expect, if your bacteria, if say E. coli is present, it will bind to the corresponding aptamer, which will cause an impedance increase, right? And this signal, electrical signal of impedance increase, will be relayed to the neural chip. And so let me just describe neural chip very briefly. So the central portion of the neural chip has a falcon head, which is the blue cylinder, as you can see. Inside this falcon is where we'll be putting all our neurons. We'll be culturing the neurons. And it is fitted with a filter because you don't want it to get contaminated. So the red sort of cap is like the filter. And the neurons we are using is N2A and SHSY5Y. These are neuroblast cells. And we put them inside, the center of the neural chip. The chip has 6 electrodes. Five are, okay. It has 6 input electrodes. 5 are visible here. There's one more electrode on that side. And it has 2 output electrodes. So the input comes in from the test strip, the aptamers on a test strip. And the output goes to a microcontroller. Right? So these electrodes are important for carrying signals to and fro from the neurons, the central portion of the neural chip. Now, the microcontroller basically interprets the output signals from the 2 output electrodes, which are coming from the neural chip, and it displays which bacteria or which microbe is present in that particular sample. So, for example, if you have a sample with E. coli and Salmonella typhimurium, then the microcontroller should display that in your sample, E. coli and Salmonella typhimurium is present. Well, the microcontroller won't display it. Is attached to some sort of small LCD screen or LED screen that will display that, okay, E. coli and Salmonella is present. If you have just E. coli, it should display just E. coli is present. If you have just Salmonella, it should display just Salmonella is present. If you have just penicillin, it should display that, okay, some sort of penicillium species is present. Right? So I hope the basic working of our project is, we could convey it well enough. If there are any questions, you can ask us. Then we'll move on to a slightly different aspect, which is the sort of the work that we have done on the project. Briefly explain that maybe. Is there any question regarding the platform, what it does, how it works? If not, we'll just... Right. And of course, you can guess they will be connected by wires and all, but yeah, these are the three principal components, right? Okay, so moving on, the work that we have done so far. Yeah, so first of all, we got access to this pretty cool machine called a vacuum deposition chamber. We used it to deposit titanium and gold onto our chip to form the electrodes. The electrodes have to have metal particles deposited, and you can see that this is how the chip looks on a petri dish. I think, can you see my cursor? Okay, great. Okay. Okay, so this is how the chip looks, right? And this is the falcon head, which we are sort of sticking onto this particular chip. And this is the BSL-2 hoods that we have access to, where we are doing our cell culture work, and we are also working with, Salmonella is a BSL-2 organism, so we have to be careful with that. We are working with all those in the different BSL-2s. And then we move on to some of the dry lab things. This is the particular simulations that we have done, which sort of try to model how the neural chip works. And I'll give it over to Kalyandeep to explain this part.
Yes, so this actually explains the low voltage, high current, and the high voltage, low current, the low frequency, and the similar sort of input parameters that we talked about before. So this is what the figure shows is basically 10 downstream neurons connected to one upstream neuron, and each of the downstream neurons are a poisson neuron, so they are firing with their own independent poisson distribution. And you can have an external input, and that external input will only have a certain frequency, and the frequency is, as mentioned, it's 10 to 50 hertz, is an increment of 10 hertz in every simulation. And what the first figure shows is that when the frequency range is around 10 hertz, all the synaptic strengths start from zero point 0.5, that is half the actual potential of the synaptic strength, but most of them fall down. So there is no synaptic strength evolution at low frequencies. However, when the voltage goes to 20 hertz, there's a competition initially at time around 3 seconds, and then two of the synapses get strengthened. They go to nearly 1 and zero point 0.8, while the others stay constant and some drop down to nearly 0. The same is observed in 30, and but in 40 we see that much higher number of neurons sort of go towards the 1 region, while much lower number of neurons go towards the 0 region. So as I was saying, by changing the external parameters, the frequency parameters, we can have a global effect in which one of the neurons, so these two neurons were not selected, the neurons that went up were not selected, it's just due to random chance that two neurons got strengthened to a synaptic strength of 1 and zero point 0.8 respectively. But we can show that we can induce global property changes by modulating the input parameters. So this will allow us to implement a kind of backpropagation in our biological system, and therefore we have basically each of the neuron learns via Hebbian plasticity, but the entire system learns using a sort of backpropagation. So it's stochastic backpropagation. Yeah.
Okay. So yeah, we are near the end. Hope you guys are not very impatient. Just let me know if I run out of time. Yeah. So another aspect is, one aspect is the neural chip, as you can guess. Another aspect is the aptamers. Right. Yeah. Okay. So this sort of gives you the different shapes of the aptamers that we are getting when we are folding them. We are using the software called M-Fold. So the aptamer for penicillin G looks like looks like this, which is pretty funny actually to look at. The aptamer for E. coli looks like Mickey Mouse. We don't really understand why, but it looks like. And the aptamer for Salmonella typhimurium looks like this when we are folding it, right? And so the thing is, one important aspect of our simulation is that since these aptamers, the one for E. coli and Salmonella typhi, have been generated using something called whole cell selects, that means that we do not know what exactly the aptamer binds to. We know that it binds to a bacteria, E. coli, but we don't know which part of E. coli it binds to. Does it bind to a protein? Does it bind to a glycoprotein? If so, which one? E. coli has a lot of surface glycoproteins. E. coli has a lot of different proteins, right? So that is one of the important aspects where we try to identify which protein the aptamers for E. coli and Salmonella typhimurium possibly bind to. So we do that using something called a protein homology search, and after the protein homology, we do something called molecular docking and then molecular dynamics to sort of find out how exactly uh, these aptamers bind. Okay. Okay, here this is a docking. There's a particular docking. So this is an aptamer. As you can see, it's actually pretty large. It's, I think, 90 bases. So this folds into this particularly weird structure. Okay, this is the aptamer for penicillin G, and this is the benzyl penicillin molecule. Okay, this tiny little green one, I think this is visible. I hope this is visible. Is the benzyl penicillin molecule, and after this docking, this is the best configuration of docking that we have. This shows that this is the particular region where the penicillin actually goes and binds to, right? And this is the data that we get from the docking, which shows you that this is the run number 10. Run number 10 has the lowest binding energy, and lowest binding energy is the most stable one. We have done this using hex dock, in case you're interested, hex docking software. And this shows you that this particular configuration has the lowest binding energy, and this is probably how the penicillin is binding to the aptamer, right? So I think with that we have more or less covered whatever we planned for today. I think we've also almost out of time. So this is the team. We have three professors from IISER Mohali, from Department of Physical Sciences, Biological Sciences, and then we have a scientist, from Dr. Raminder Sundari from Institute of Nanoscience and Technology, which is just beside our campus, who's also helping us with the Aptamer experiments. We have our instructors who are basically PhD students working in INST and IISER, and we also have a guy from ISI Kolkata, and we have advisors who are basically last year participants of iGEM, and we have the core team, which are the student members, which is me, Kalyandeep, and even Prerna has joined the meet. And we have, these are the people who make the project possible. And I would also like to thank the various volunteers. The human practices especially requires a lot of different volunteers, and who have really helped us make the project a success so far. So thank you guys. If you have any feedback, any sort of questions, please go ahead and ask. I think we have maybe 5-10 minutes left.